Thank you, Jonathan. It's a pleasure to be back here at Edinburgh again, a place I love very much. So thank you for the invitation. And as Jonathan said, it's a tale of two cities, really, that involves Oxford, where I started out, Bristol, where I then went to, and Oxford, where I went back to. So Oxford Ragwort, Bristol Whitebeam are going to feature in my talk this evening. And it's really about hybridization in plants and what that can lead to. The early exponents of neo-Darwinist thinking, population geneticists like Fisher, were, let's say, very zoo-centric in their thinking, and that statement from Fisher about his view of hybridization there, the grossest blunder in sexual preference which we can conceive of an animal making. Animal is the key thing there, because there was a lot of animal zoo-centric thinking going on at that time. And of course, that was behind the thinking behind Ernst Mayr's biological species concept, which no doubt many of you will be familiar with. And he went on to say hybridization is an unnatural breakdown of isolating mechanisms, but he hadn't really factored in what plants get up to, particularly in their reproductive behavior. And it was left to great plant evolutionary thinkers, biologists like Ledyard Stebbings, and then later Vern Grant, to point out that animals, that plants are more problematic than animals because they can hybridize, and many will self-fertilize. Others will have various forms of asexual reproduction, from clonal reproduction to seeds without sex, agamospermy, popularly called now apomixis, more generally, and of course polyploidy, which is associated with hybridization, asexual reproduction as well, to create difficulties when it comes to defining species and classifying, categorizing taxonomic entities in plants. And this wonderful book came along, Vern Grant's Plant Speciation, in which he devoted no less than 6 chapters to natural hybridization in plants and its products. And then in more recent years, the likes of Lauren Rieseberg have taken this hybridization work into the realms of genetics, molecular genetics, evolutionary genetics. And on the animal side, people, particularly Jim Mallet, with his work on hybridization in heliconius butterflies, has shown that hybridization has actually played a creative role in animal evolution as well as in plant evolution. So it's no longer perceived by evolutionary biologists as an evolutionary dead end, which Fisher and Grant and others saw it as. And evolutionary consequences of hybridization in plants can be quite varied. And You can get F1 hybrids, which are often sterile. They can be stabilized through asexual reproduction, apomictic lineages, clonal lineages, and then gene duplication, leading to allopolyploids, can then lead to speciation. Then up here you can get fertility, progressive fertility in F1s and subsequent generations, leading to perhaps ecological diversification, so-called transgressive segregation of genes and alleles that happens in sunflower, introgression, which we'll see a bit later. But the main part of my talk is going to be focusing on this area of hybrid speciation in the form of homoploid hybrid speciation, where you get hybrids forming without a significant, well, without polyploidy, without a change in the chromosome number. So typically fairly closely genetically related species forming partially fertile hybrids that then go on to be fertile and reproductively or geographically or ecologically separated from their parents. And then allopolyploid speciation, where you get, say, usually a sterile triploid that then undergoes a spontaneous doubling of the chromosome number to, say, become an allohexaploid, and that restores fertility, and also it separates that lineage from crossing with its parental diploid and tetraploid parent. And then you get the agamic complexes that can arise as well, and so-called microspeciation, which leads to incredible taxonomic difficulty. Think of blackberries, for instance, rubus. There are innumerable different microspecies of blackberries that, and the blackberries you eat are all derived from asexual fruit set, seed set, without normal sexual processes. This is the classic now textbook example of homoploid hybrid speciation that was pioneered by Lauren Rieseberg, working on sunflowers. These two North American sunflowers, Helianthus annuus, that grows on mesic clays, seasonally wet, hybridized with Helianthus petiolaris, which grows on open, dry, sandy soils, to create these three new lineages that have totally different ecological preferences to the two parents. So Helianthus anomalus grows in sand dunes, adapted to very arid and dry conditions. Similarly, deserticola growing in arid deserts, and Helianthus paradoxus that grows in salt marshes. And one of the great things that Rieseberg and his colleagues did in these studies was recreate these hybrids synthetically by doing crosses and planting out many thousands of F1s, F2s, F3s in these three different environments and selecting for the progeny. So he got these not through his own means and manipulations. So recapitulating what thousands of years of evolution had selected for in the original hybridization process. More common is allopolyploid speciation, and there are numerous examples. A lot of our agricultural crops are allopolyploids, and it may be a perception that this is more common because it's easier to detect than footprints of homoploid speciation. So there may be more homoploid hybrid examples of hybrid speciation around, but it's more difficult to detect. So there are familiar examples there. Oilseed rape is an allopolyploid, and there are various cereals that are allopolyploids, as well as Senecio cambrensis, which had a second origin up here in Edinburgh, now I believe is extinct in Edinburgh, normally called the Welsh ragwort, as in cambrensis. So I'm going to move on to Oxford now, and the first of the eponymous plants, Oxford Ragwort, which is its history is intrinsically associated with Mount Etna in Sicily, and it's a story really that was championed through this wonderful work by George Claridge Druce, The Flora of Oxfordshire, that went through a number of editions, but in the 1927 edition he really waxed lyrical about the origin of Oxford Ragwort, and he collected lots of accounts from others that had deduced through various historical writings that Oxford Ragwort was introduced to the UK from Sicily, and specifically from Mount Etna. And that introduction was around 1690 to 1710. And it was introduced to the Oxford Botanic Garden at that time, probably via seed exchanges from botanically orientated monks on Sicily, and the then keeper of the Botanic Garden, Jacob Bobart the Younger. And he really goes to town on telling this story, and that is really where the whole thinking of the Oxford Ragwort's origin on Mount Etna came from. And then he waxes lyrical about how the clinker beds of the railways that were expanding all over the UK during the Industrial Revolution created the same sort of environment to what it was used to growing on Mount Etna. And these railway lines and more recently roads and during and after the Second World War, bomb sites became the favoured habitats of this plant. And having escaped from the Botanic Garden, it sort of sat there in the Botanic Garden for about 100 years, and then started to grow on the crumbling walls of the colleges and the university, made its way to the new station, and then kicked off along the railway tracks all over the country. And you can see it still today. brightening up railway lines all over the country. There are very few railway stations in this country where you can't see Oxford Ragwort at its two peaks of flowering in May and in September. And there we are. I mean, I took that right at the start of when I began my research into Oxford Ragwort back in the early 1990s, just as they were rebuilding Oxford Station. And there we are, the clinker beds of the railway lines furnished the plant with a replica of the lava soils of its native home in Sicily. And this spread is really well documented in the literature of local botanical societies. And that gives a real chronology of this incredibly rapid spread. And this is an invasive that is really, I see it as a bright, non-destructive. It's not like Japanese knotweed or anything like that. It brightens up these gloomy urban man-made sites, bomb sites, building sites, derelict sites. Still in Bristol, there are old bomb sites of the city that still haven't been built on, and you can find Oxford Ragwort along with buddleia and willow herb, which all flourished during the war and after the war on these old bomb sites, just brightening up the whole environment. So that it spread really rapidly and covered most of the British Isles in about 150 years, which is quite something. And then Druce goes on waxing even more lyrical about getting on a train in Oxford with an akeen of Oxford Ragwort, and then it alighting with him in Tilehurst. You know, he's really going to town on this story. And many of you who are familiar with the work of the late great Richard Abbott, spent most of his life at St Andrews working on Senecio, will be familiar with the progenies of a hybridization between Oxford Ragwort and our very much native Senecio vulgaris common groundsel, which is a tetraploid. And the cross generally works this way with vulgaris as principally the female component, and they produce this a sterile triploid hybrid, nominally known as Senecio times Baxteri. And this has led to 3 new taxa appearing in the British flora since the 1800s. The first to arrive was an introgressed form of the common groundsel that has the ray flowers, ray florets of Oxford Ragwort. So it's inherited the genes that make these petaloid flowers and around the edges. And that's a tetraploid. So it's possible that that this is a reduced gamete, a normal 20x female gamete with an unreduced male gamete of 20x from Squalidus, creating that. And that was first recorded in 1832. And Richard Abbott did a lot of work on this, particularly showing that the presence of these more showy ray petals increased the outcrossing frequency of this, because this is a habitual selfer. It's got all the features of a very prolific selfing plant, but this is more outcrossed, simply because of having those showy ray flowers. And then came in 1948, first recorded in Wales, the hexaploid derivative of this, Senecio cambrensis, at 6x. And then Richard described in 1979 the York ragwort, Senecio eboricensis, which is another tetraploid, but seems more more like Oxford Ragwort in its stature than Hibernicus, and that was given species status by Richard and others in the 1990s, when they fully described it and showed that it was a species. So a lot has happened with the presence of Oxford Ragwort in the UK. 3 new taxa originating from that. And one of the areas that I got interested in when I'd sort of drifted away a little bit from working on the reproductive biology of Oxford Ragwort, I was mainly focused on looking for the genes involved in self-incompatibility that creates its obligate outbreeding system. I got interested talking to Richard about this particular plant, and it was at a time when genomics and transcriptomics, as it's called, the study of RNAs and the synthesis of the protein templates from DNA that were then people were able to analyze at large scale. And we used transcriptomics in terms of microarrays that were just being really developed and utilized in the early 1990s. They developed, and we started using them in the early 2000s to look at gene expression in resynthesized lines of these. So one of the beautiful things about Senecio cambrensis is you can recreate it relatively easily by crossing. You have to emasculate, which is not an easy task with groundsel. You emasculate it to make sure it can't self-pollinate and self-fertilize. And then you pollinate it with pollen from Senecio squalidus. You get the triploid hybrid. You track these, verify this with flow cytometry, which tells you what the ploidy level is, what the chromosome number is. Treat with colchicine, and that will double the chromosome number, and you can get synthetic lines of cambrensis and take them through different generations. What is interesting is that immediately that you double up the chromosome number, you get restoration of self-compatibility. So they're fully fertile. And when you create the hybrid, Senecio baxteri, you get what we described as transcriptomic shock, where the expression of genes from both the parents is sort of exploding and going in all directions. And when you treat with colchicine, and this was quite a revelation that we discovered, many hundreds of the genes will suddenly, in the first generation of hexaploids, start to go towards that seen in the wild Senecio cambrensis, and you get almost to that point after 5 or 6 generations of tracking the hybrids. But that's a lot of work that I'm not going to necessarily dwell on, but it was something that was very productive for us in the early to mid 2000s. So what about the origin of Oxford Ragwort itself? I started off with the Druce account, the Oxford Ragwort story, where he categorically said that it came from Sicily, and that story has continued through other people's works who quote Druce. So Edward Salisbury's famous book on weeds and aliens, and more recently Richard Maybe's book on weeds, all saying came from Mount Etna in Sicily. And yes, it did. Well, its parents grow on Mount Etna. We know that. The two parents are Senecio chrysanthemifolius, which grows at low elevations on Etna, typically below 700 meters, and Senecio etnensis, which grows at high elevations, typically above 2,000 meters. And at points on the volcano, they get together, and there's a hybrid zone, which has been the taxon there has been described as Senecio incissus and various other names if you look in herbaria in Italy. And it's thought that it's that hybrid that is what our Oxford Ragwort Senecio squalidus is derived from. And it's a homoploid hybrid, just like those sunflowers that I started off with. And we know from molecular marker work that it definitely does is a hybrid between these. Richard Abbott, with a PhD student in the 2000s, used molecular markers to show that it is a mix of Senecio chrysanthemifolius and Senecio etnensis, approximately 60 to 70% chrysanthemifolius, 30% etnensis. This is British stock. So it's definitely a hybrid between these two Mount Etna natives. And when we did some more intense molecular marker analysis, this was back in 2013, what we noticed was that it these molecular markers didn't cluster with the Mount Etna taxa, the parents and the hybrid, which, as you can see, hybrid from Mount Etna sort of overlaps quite clearly with both the parents, but Oxford Ragwort sits to one side. When we looked in more detail with looking at the genomes through the RNA profiles with many thousands of genes, this was even more pronounced, that Oxford Ragwort sat quite away from both the parents. Interestingly, the hybrid on Etna sort of moves toward, it overlaps more with chrysanthemifolius parent than it does with etnensis. So there's something a little bit strange about Oxford Ragwort. And so we sort of thought, did it actually come from hybrids on Mount Etna? What's the evidence in the herbaria to say that we had hybrids in Oxford? In Oxford, there's clearly herbarium evidence of what looks like a hybrid between the two parents. And certainly there's a lot of herbarium specimens of one of the parents, Senecio chrysanthemifolius. But Bobart was exchanging plants with the Duchess of Beaufort down at Badminton in Gloucester. And when we looked at her herbarium, lo and behold, we found both parents were growing in her garden at Badminton. So we thought, what if the hybrid had formed at Badminton rather than on Mount Etna? And to cut a long story short, we tested this hypothesis with lots of sequencing of all the transcribed genes, the transcriptome. We sampled both parents, the hybrid on Etna and Oxford Ragwort in the UK, and then used this sequence polymorphism data with one of the experts in a technique called approximate Bayesian computation, which is a statistical method. I don't understand it myself, but that's why we got Beaumont to do it. And he used these data to test the hypothesis as to whether the origin was more recent, i.e. a few hundred years, or more ancient, which it would have been if the hybrid, because the hybrid's been forming on Mount Etna for many, many thousands of years. And this landscape essentially is a probability landscape, and the highest probability is where it's showing yellow, and where the data, the observed data sits in this landscape is where the highest probability is. And if you look at the red dots here for a more recent origin, then the probability is far higher than for an ancient origin using the Oxford Ragwort data from the UK. So we have to revise our thinking about Oxford Ragwort now in the light of 95% probability that it arose within a few hundred years as a hybrid in the UK. And therefore, perhaps the Oxford Ragwort is actually a Gloucester Ragwort. So I'll leave you with that thought and move to Bristol. So when I moved to Bristol, having been in Oxford, I quickly found out with conversations with local botanists and in the area that there was a Bristol Whitebeam, a red data listed plant growing in the Avon Gorge, along with many others of endangered and endemic whitebeams of the Avon Gorge. And here, it's very common. It is the sort of diversity hotspot of whitebeams, of Sorbus in the UK. And there are 3 sexual species, including rowan, common whitebeam, and wild service tree. There are 6 native polyploid apomictics. Some are triploid, some are tetraploid. There's an F1 hybrid. There are some introduced ones. And importantly, the local experts had identified through morphological analysis some oddities that were neither one or other of these other taxa. And so we got some money from the Leverhulme Trust and NERC to look more closely at the relationships between these all these different Sorbus in the Avon Gorge. And that's my colleague Tim Rich. Some of you will be familiar with him. Not always easy to sample Sorbus in the gorge, but he and another colleague were very familiar with the Sorbus in the Avon Gorge, and we set out with the aims of determining the pattern and processes of evolution of Sorbus in the gorge, confirm which species are hybrids, polyploids, apomixis, and ultimately to use the outcome of this research to make recommendations for the conservation of Sorbus in the gorge, because conservation and management processes were focused on conserving the red data, the highly rare endemics that were only found in the gorge. So we wanted to look at that way of working as well. These are other people that were important in the work. Some of you may have heard of Ashley Robinson, who worked with Richard Ennos up here in Edinburgh on the Arran Sorbus, the speciation that has gone on there. And he was the postdoc on this project and did most of the molecular work. And then Libby Houston, who is a local expert, knows the Avon Gorge better than anyone else around, and has an eye for Sorbus and spotting oddities, and saying you must sequence this and see if it's different, which we tended to do based on her insight. And then Shana, who did some of the work that I'll talk about at the end. We used molecular markers, microsatellites, repeat sequences that are very diverse in closely related taxa to get an idea of the profiles of the different Sorbus species, microspecies, taxa, whatever you want to call them, in the Avon Gorge, and develop profiles, molecular profiles for each of the different taxa. And we used these data to address a number of questions, particularly the relationships and the origins of the polyploids, and particularly the triploids. And this table here, if you focus on the number of individuals here, Ni, you see quite a lot sampled up here of the diploid sexual taxa, and the number of genotypes over here reflecting the genetic diversity. So it's very high. Each one of these individuals was genetically different. So very high genetic diversity. Over here with the triploids and polyploids, tetraploids mainly, you can see there's quite a lot of individuals. There's only one individual of this strange eminens type. There are a number of individuals sampled, but generally only one genotype was identified, showing that they're all clonal, apomictically reproduced. And there's Sorbus bristoliensis. We sequenced 32 individuals, and they all had exactly the same genotype, which shows that there was a single origin, they're all genetically identical in the Avon Gorge. And by comparing the profiles, the microsatellite profiles of the different Sorbus, we could work out who the parents of the triploids and some of the tetraploids were. So here, the profile of Bristol Whitebeam, Sorbus bristoliensis, is basically markers from Sorbus eminens, which is a tetraploid, and Sorbus terminalis, which is a diploid. So tetraploid plus diploid will give you a triploid. And the profiles were all identical, showing that that's the likely hybridization that led to Bristol Whitebeam. And we did that for plenty more. So there's the parents of Bristol Whitebeam, eminens, so-called round-leafed whitebeam, and terminalis, the wild service tree, which is generally the female in that cross. And we did another rare one, Wilmot's whitebeam, Sorbus wilmotiana, and that is a hybrid of Sorbus aria, the diploid, and Sorbus poragentiformis, the tetraploid, etc., etc. And this is one of Tim Rich's doodles, the Sorbus family tree, where he's used all common names. Tim was he still is, he's a wonderful taxonomist, and he gets quite obsessed with these agamic complexes and giving them all names. So we've got whitebeams named after most of the people who were involved in the project. He never named one after me, which I'm a little bit peeved about, but maybe they will discover it. So there's Sorbus times Robert Stoney, named after Ashley. There's Sorbus times Houstonii, named after Libby Houston, and others. But we confirmed that, we're only showing 5 of the oddities there. We confirmed that all 6 of the oddities identified by Libby Houston were genetically distinct and derived from particular hybridizations between diploids and tetraploids, or derived tetraploids and derived triploids in the gorge. And since that work was published, I think Libby's identified another 5 that have gone on to be looked at and shown to be genetically distinct. So it's like a sort of great amoeba of a gene pool that is budding off into all these different forms that either become established or not in this ecologically very diverse environment of a gorge. And so evolution is ongoing. That's the key point here, that these new taxa are constantly being created through some sexual reproduction and then frozen with asexual reproduction. So you're freezing a particular new genotype that will be either selected for in a particular niche, or it won't be selected. It won't find a niche that where it's at an advantage, and it will become extinct. So this is a term that was coined way back in the 60s and 70s. It's a syngamion of different genotypes and forms within a gene pool. So what is driving this process? And this is where Shana comes in with her work. And we did controlled cross-pollinations, controlled self-pollinations among various different Sorbus in the Avon Gorge, and looked at what was compatible, what was incompatible in terms of pollen tube growth in the pistil and fruit set, seed set afterwards. And the other thing we did was we separated, this was a technique that Ashley perfected, of separating the embryo from the endosperm, because we weren't sure on what the ploidy of the endosperm was in these triploids. And so it was possible to separate out the embryo and the endosperm and then genotype both of those separately. In terms of the crossing study, what Shana showed was very conclusive, and it showed, not surprisingly, in the diploids, which are self-incompatible, that if you self them, you don't get any seed or pollen tube growth, but if you cross them, you get seed. However, in the triploids, including Sorbus bristoliensis, you self them, you don't get any seed or fruit. You cross them, you don't get any seed or fruit, which is not surprising because they're all the same genotype, because they've all been derived from a single cross. So the self-incompatibility is still working. But if you cross them specifically into taxon-wise with a diploid or a tetraploid, then they're compatible. And the tetraploids are all self-compatible, and they're all cross-compatible, and they're all compatible with a diploid. So what this shows is that that so-called pseudogamous seed set, because even though the embryo is not formed from sexual fertilization, it's a clone of the female, the mother, the endosperm that nourishes the embryo in the seed is actually outcrossed, and you have to have cross-pollination to get the endosperm. So you can't self-pollinate an agamous triploid and get an endosperm, because and that means you won't get fertile, you won't get seeds. So this is interspecific or intertaxon pollination driving this whole process. And that was quite a discovery. And when you then look at the endosperm, and genotyping the endosperm, you find that the endosperm is very genetically diverse compared to the embryo, which is all the same. We don't know yet what the implications of outcrossed endosperm is in this process, and we didn't get the funding to pursue it either. So we're still speculating on what the consequences of this may be for perhaps fitter seeds or better offspring success on germination. And we thought, is this a one-off? Is this a sort of, is this unique to the Avon Gorge? And having confirmed a lot of what we set out to do in the first place, we speculated as to whether limestone outcrops and valleys, gorges, they provide multiple novel niches. in the gorge, and that's probably what's allowing these new forms to establish, like this crazy thing that is growing on near vertical cliffs, where other sorbus can't get a foothold on. That's actually Sorbus times Robertstonii, which is very much threatened simply because of rock slides and other events that put its niche at risk. But we asked, are there other examples of this going on? And looking in the literature, of course there are in the UK, in the Cheddar Gorge particularly. There's a lot of Sorbus variation on Exmoor and in the Wye Valley, and of course on Arran. All of these are limestone outcrops, and across Europe, into Eastern Europe, limestone gorges and valleys are all hotspots of Sorbus diversification. And this is the story that was articulated by colleagues at Exeter University, who we worked with to basically do a similar study on the Exmoor Sorbus as we did on the ones in the Avon Gorge, and particularly looking at this famous no parking whitebeam described by Mike Proctor, Sorbus ad monitor. So it had a no parking sign nailed on its trunk, and that was found to be an oddity that had been created by this process with the Sorbus there, again focusing around crossing between tetraploids and diploids in the form of Sorbus terminalis and Sorbus margaretiae. And across Europe, even more diversity in the Dija, if that's the way you pronounce it, valley in the Czech Republic. Martin Lepsky had been categorizing these things, and went on to use our techniques to confirm that similar processes are going on there as well. And also in Hungary, Saba Nemeth is doing the same, and has shown similar processes going on there. So in the short of it, these processes are going on wherever there are limestone gorges and valleys, and wherever there are Sorbus diploids and tetraploids. And going back to the question of the conservation and site management of places like the Avon Gorge, we were able to make recommendations to Natural England and the Avon Gorge and Downs Wildlife Project that have all been implemented now in terms of preserving the process rather than the individual outcomes of the rare and endemic forms. So now all of Sorbus is being conserved and preserved in the Avon Gorge to allow this evolutionary process to continue, because inevitably many will become extinct to do with climate change, and new ones will evolve to fill their place. And there is Libby again, who is still carrying on even well into her 80s now to champion the cause of Sorbus conservation and make sure that the Avon Gorge and Downs project do what they should be doing in terms of conserving this amazing process. So sorry I've gone a little bit over, but thank you all for for listening.